I'll start sharing my screen now, and then we will start with the session in just a few minutes. I hope you can see my screen now, right, Namita?
Yes, yes.
Okay, thank you. Okay, then. So without further delay, I'll quickly start with the session in that case. So, hello everyone, a very warm greeting for today's webinar on From Classroom to Cancer Research, a Biotechnology Student's Journey into Bioinformatics. So, this is a student project presentation webinar featuring Namita Biju, who is a MS Biotechnology student at the University of Queensland and an alumna of OmicsLogic Biomedical Data Science Research Program. So, for those participants who are not familiar with the program, I'll be covering more about it in the upcoming slides. But just as a quick objective of the today's webinar, so in this webinar, Namita will share insights from her independent research project on understanding gene expression and resistance mechanism in non-small cell lung cancer. And we will also be going through her exploration of gene expression data in non-small cell lung cancer and highlight the biological insights that she has uncovered through her project. And at the end of the session, we also have some exciting research opportunities that participants can pursue, and we'll also discuss some exclusive scholarship opportunities that the webinar attendees can avail. So before we begin, for those participants who have joined with us for the first time, let me take a brief moment to introduce you to the team behind the webinar. So, we are a global bioinformatics and data science company with roots in India and the US. And over the years, we have had the privilege to collaborate with leading universities, research institutions, and industry partners worldwide. And our mission is to bridge the gap between data driven research and skill development. To achieve this, we work with regional and international experts who help us tailor our program to the specific needs of students, educators, and researchers across the globe. Now, with the growing demand for skilled bioinformaticians and data scientists, we have developed a range of training and research offerings. These include online courses, workshops, internships, and fellowship, research services, and even a degree program. Now, here's the overview of the student learning journey that begins with hands on courses or programs that is suitable for all levels. So, even if you're a beginner or even if you're an advanced learner. And for those looking to go beyond, we also conduct the research program that the Namita was also part of, so which provides a platform to work on real world projects with expert mentorship. And here's a quick glimpse into some of the courses and example projects that we offer on a wide range of topics from precision medicine, oncology, infectious diseases, metabolic disorders, to even agri-bioinformatics. Now, all of our courses are enriched with engaging content, including visuals, infographics, and video lectures. And we also heavily focus on experiential learning through hands on practical demonstrations and coding exercises. And while our courses are fully automated and lesson based, we also understand the value of mentor guided instruction. So that is why we also offer workshops and training programs that combine instructor led sessions with hands on walkthroughs to reinforce key concepts. So this also includes a combination of expert video lectures, interactive tutorials, and step-by-step lessons. Now, this is the research program that I was talking about at the beginning of the webinar. So, for those of you who are not familiar with the program, it is an unparalleled opportunity for students to work alongside a mentor on a topic of their choice, and they could choose to specialize in a specific omics field like genomics, metagenomics, transcriptomics, cheminformatics, metabolomics. And once they specialize and they complete their training, they can choose to work on a research project on a topic of their choice. So these could be on infectious diseases, agriculture, precision medicine, metabolic disorders, neuroscience, oncology, immunology, and so on. And here's a quick glimpse into some of the previous preprints and publications that have been carried out by the students from our previous research program. So these are on topics on diverse range from stem cell biology, space medicine, cancer research was also one of the most popular among the students. And all of these preprints and publications and research projects, they have been undertaken as a part of different objectives, like students want to get research experience, some of the students want to showcase their research work as a part of their higher studies applications to universities. Some of them also want to press and post at presentation competitions and so on as well. So these are some of the areas that we have done. And one important highlight of this preprint is that this was completed by a high school student as a part of his interest in completing a poster presentation competition. And these are some of the feedback that we have received over the years about the programs that we conduct. Now I'll just quickly stop sharing and reshare my screen again and take you to the OmicsLogic Learn platform if you're new to the platform and would like to explore some of the resources that have been covered by the students through the previous programs. I'll just quickly take you to the platform. So this is the platform. I'll share the link in the chat box for those participants who are yet to sign up. So very simple process to creating an account. You can simply head over to login and you can click on create an account. You can enter your name, email ID, password, and you can create an account. Or you can also sign up using any of your existing social media handles, such as Google, Facebook, Apple account, GitHub account, or even Twitter. Now, I already have an account on the platform using my Google account, so I'll log in using that. And once you sign in, the very first interface that you see is the courses page. So here we have a diverse range of topics that I was covering in my slides as well. We can explore the various coursework on topics such as bioinformatics, genomics, transcriptomics, machine learning, Python, R, metagenomics, molecular modeling and cheminformatics, single cell, epigenomics, Linux, and GitHub. Also, if you have any specific interest that you are interested to explore, you can simply enter the keyword that you're interested to learn, for example, transcriptomics, and it will show up the different coursework and example projects that cover such topics. And you can explore all of these at your own pace as well. I'll just quickly open up one of the coursework just to show you the format. So here is the example of transcriptomics, one of the most popular coursework among the participants. And as shown in the slides, they are all detailed and comprehensive. And there are step-by-step instructions wherever there are hands-on analysis. There are instructions on how to perform the analysis, the required softwares to be installed, and also the data set that is being worked with, and so on. And there are also GIFs, screenshots of the various outputs that you get and how to interpret them. All of that are also covered within the coursework. So this is just a quick overview that I wanted to give you about some of the study resources if any of the participants are interested to explore through the OmicsLogic platform. Now, without further delay, I'll pass over the stage to the speaker for today's session, Namita Biju. Over to you, Namita.
Thank you so much.
Thank you.
Hi, my name is Namita, and I have completed my bachelor's in biotechnology, chemistry, and botany, and right now pursuing a master's in biotechnology at the University of Queensland. So for me, right now I'm concentrating more on the advanced biomedical applications, which includes molecular diagnostics and therapeutic strategies. And during my bachelor's, I realized that modern biology is no longer about wet lab experiments. It's more of a combination of next generation sequencing techniques and computational tools. So that's what motivated me to actually transition from a purely biotechnology background into a bioinformatics to make sense of the various data sets and about more about the computational levels. So of course it wasn't easy, especially coming from a pure lab-based background. So at first, coding in R and Python was a little bit difficult and daunting. But slowly, I think I got the hang of it, especially with the help of my mentor, um, Ritika Patyal. She was very helpful in providing her guidance during entirety of my student project. So yeah, it's a very supportive environment, and it really helped me bridge the gap between the wet lab techniques I learned and also the bioinformatics. So in the omics, related to the omics program, I enrolled in the transcriptomics beginner program. So before that, I attended a 7-day workshop for big proteomics. And after that, I enrolled in the research program, which was really helpful and gave me a lot of insights and background and really helped me provide a research component while applying for the university. So yeah, thank you for having me. And yeah, so my student project the topic was on the gene expression in non-small cell lung cancer. So some of the key insights would be that non-small cell lung cancer is a very what do you call it? A very daunting topic since it has almost 85% of cancer cases and also 1.8 million deaths annually. And the problem is the late diagnosis for it that so due to its similarity with common illnesses. And key factors for risk include smoking, air pollution, occupational hazards. So it makes it much more surprisingly common. So the symptoms of non-small cell lung cancer, as you can see, are very similar and vague, like the persistent cough, shortness of breath, chest pain. All these are overlap with various common illnesses. So it is diagnosed very late, which is very detrimental to the patient. Um, so it is classified into three subtypes, the adenocarcinoma, squamous cell carcinoma, and large cell carcinoma, where the adenocarcinoma is one of the most prevalent types, and it's often associated with EGFR and KRAS ALK mutations, and LUSC associated with the TP53 mutations. And large cell carcinoma being the most undifferentiated and aggressive one. So, concentrating on the EGFR signaling, it plays a central role in other non-small cell lung cancer, especially in relation to adenocarcinoma, where it is a receptor kinase, tyrosine kinase. So, when it is activated, it will trigger downstream signaling that promotes the proliferation, survival, angiogenesis, migration of the tumor cells. And when in NCLC, the EGFR mutations, such as the exon 19 deletions or the AL858R point mutation, exon 21, there is uncontrolled tumor growth, which is very bad and detrimental. So the tumors would respond to this tyrosine kinase inhibitors, like examples are gefitinib, erlotinib, and osimertinib that blocks the receptor. So it can induce regression. But resistance is very common and it often emerges within a year of treatment. So mechanisms like secondary EGFR mutations, activation of bypass pathways, all these are various resistance mechanisms. So we need a new method which combines strategies and continuous molecular monitoring for long-term management. So we have looked at the biology and the clinical features of NCLC. So over the last 15 years, targeted therapies like EGFR inhibitors, as mentioned before, has changed how we treat the adenocarcinoma especially. So people who could survive for a few months can survive for a longer time if we identify the right mutation. But the bad news is that it has resistance, and almost every patient develops resistance within 9 to 14 months, no matter what generation of the inhibitor they are on. So, and NCLC isn't just a signal disease. Adenocarcinoma, squamous cell carcinoma, large cell carcinoma have very different molecular drivers. Even within adenocarcinoma, the two patients with the same EGFR mutation responds differently. So that means there are hidden molecular features at the gene expression and pathway level, which we don't fully understand. So the big picture here is that the treatment has made progress for NCLC, but resistance and subtype heterogeneity is a huge unsolved problem. So we need a systematic comparison of how the transcriptomic landscape will differ between tumor and normal lungs across the different subtypes, especially in the context of EGFR signaling. So that's what we have done here with the objectives of first profiling the transcriptomic differences between the tumors and normal lung tissues. And then we identify the differentially expressed genes and pathways with special attention for EGFR signaling. And then we highlight the potential biomarkers and therapeutic targets to guide the personalized treatment in NCLC. So finally, our study will focus on the transcriptomic differences between LUAD and LUSC mainly, and we hypothesize that each subtype has a distinct gene expression signature and pathways. So by analyzing the public data sets, we aim to identify biomarkers and therapeutic targets to make a more personalized treatment. So this is the data set I had used, which is GSE 18842 from the GEO database. So this would have 91 total tissue samples for lung, with 46 being tumor and 45 being the normal control. And it is also from the similar patients, which helps compare it more better. And it has been validated by the qPCR, so it is reliable for biomarker discovery. So this is my methodology, where in my workflow, we first retrieved the data, then we pre-processed it with log2 transformation and exploratory analysis. And then we did PCA, which showed that there was clear separation between the tumor and normal samples, then performed differential expression analysis using DSEQ and LIMA, with filtering at adjusted p-value being less than 0.05 and log2 fold chain being more than 1. Then significant genes were mapped to gene symbols and IDs, then followed by the functional and pathway enrichment analysis using ILNX and Enricher. So this will help us identify key biological processes and signaling pathways. So in the next slide, we can see here that there's the dot plot. So in the dot plot, we had we saw the most important ones such as regulation of cell-cell adhesion and the positive epithelial cell proliferation, wound healing were all the major points noticed. Then in our PCA plot, we could see a clear separation as mentioned before, where the PC1 will have 66% of the variants, which shows that the strong transcriptomic differences, which can validate our dataset and analysis pipeline. Then we also generated a volcano plot, which summarizes all the genes analyzed. And the highly significant and strongly differentiated and expressed genes are AGER, SFTPC, and CLDN18, which we identified as potential biomarkers for NCLC. And we also generate a heat map, so we can see the top 10 upregulated and downregulated gene, where the tumor and control groups are clustered separately, and genes like SFTPC and CLDN18 elevated in tumors, and genes like GNG4 and HYOU1 are more expressed in controls. So this shows a distinct molecular signatures between tumor and normal tissues. Then we use the ILNX platform and Enricher, where KEGG pathway analysis was first done to highlight dysregulated cell cycle and p53 signaling, which is consistent with the tumor biology. And other enriched pathways include the senescence and drug metabolism. And in terms of molecular function, microtubule binding and kinase regulation were also standing out, which also contributes to cell division and the cancer progression. So the final results were that several genes are linked to EGFR signaling. So SFTPC, CLD, and 18 will affect the epithelial integrity, while RNF7, HYOU1 will promote immune evasion and stress survival. And genes like SLC25A1 and FABP4 support the metabolic rewiring and enabling resistance to the EGFR inhibitors. So these all are potential therapeutic targets. Thus, our future directions would be that biomarkers like SFTPC and CLDN18 should be clinically validated, and resistance associated genes like RNF7 and SLC25A1 also are new therapeutic targets. Then we can have isoform specific studies using CLD and 18.1 and 18.2 to reveal subtype specific therapies. And we can also integrate more dynamic monitoring for real time personalized treatments. So, this highlights very promising avenues to improve precision medicine in NCLC. So I would like to give my heartfelt gratitude to the OmicsLogic team, especially my mentor Ritika Patyal, and also thanks to Gauri Krishna Kumar and Mohit sir for all their help and guidance in especially completing this project. Thank you so much.
Thank you so much, Namita. So, if participants have any questions that they want to ask Namita, you can post it in the chat box. But in the meanwhile, let me now share my screen while the participants, if you have any questions, they are sharing. So, we do have an exciting set of ongoing research projects that the participants can join. And for those participants who are joining with us for today's webinar, you are eligible to apply for the scholarship for the month of October. So I'll share the link in the chat box to this form. So if you're interested in participating in this research program and work alongside a mentor, so if it is a part of your master dissertation or to get some internship research experience, or you want to carry out research publication or preprints, or you want to undertake a research project to present at poster presentation competitions, or any other reasons that you want to undertake a research project in bioinformatics and data science, so this program will be suitable for you. And I've shared the link in the chat box to this specific scholarship form. And there are basic details that you can enter. You can enter your email ID, full name. Just make sure that the contact details that you enter here are correct, because the team will be evaluating based on the responses that you enter here in the form. And also the contact number that you prefer the team connects with you. You can enter a few other details such as your country, company or organization or university. Mention your level of education. Are you a high school student or an undergraduate, master's, or working professional? You can mention that. And do you have a prior experience in research? So you can mention that also, so that it helps understand the team to understand your background. And also the research project, how would you like to undertake it? Do you wish to undertake it as a part of an independent project, or would you like to work in groups with your peers or your friends? You can mention that as well. Also mention the number of students if you're interested in working on group research, the number of students you're interested to work with, like 2, 3, 4. You can mention the number of students. And also for what duration do you wish to work on this research project? Is it 3, 4 months? You can mention that, or any other longer duration. We also have participants who enroll for like 6, 9 months of research work. So you could mention that as well. And also your motivation for undertaking this research project. So such as, like I mentioned in the beginning, like university dissertation or thesis or publication, or to get some research experience, you can mention that. And also just a few lines about why we should consider selecting you for the scholarship. You can mention that as well, but it is completely optional. And also a section to upload your CV. So this will help the team to assess your background. Like, are you a complete beginner in bioinformatics, or do you have some previous experience in bioinformatics and research? So you can mention that as well. So these two last sections are completely optional. If you're interested, you could fill it out. But then yes, so we will keep this form open for a few days. So just make sure that you fill it out before the timeline is over. And also, if you have any other peers or friends that you think this opportunity would be suitable, you could share this with them as well, and help them to get interested in this program. So that is all that we wanted to cover in this webinar, just to help participants get familiar with the research program, also to get an opportunity for them to interact live with the student who has already undertaken research project, and also just to understand what were the insights that she has uncovered, and also just a little bit about her learning journey into bioinformatics. So for those of you who are watching this webinar through the recording, we will also email you the recording link, and also share with you the link to the scholarship form, so that you can have all of these links in one place. And of course, if there are still some lingering questions about the research program that you have and that you would like to discuss a little bit more, you can definitely reach out to our team, Our team will share the contact details in the chat box. If you're already connected to our team members, then you can simply drop them a message with your queries and our team will be able to assist you. But if you have not connected yet, we'll share the contact details in the chat box and you can stay connected. We are also available on WhatsApp, so you can drop a message there as well. So do we have any questions from the participants?
Hello.
Yes, go ahead.
I have a question. For the team project, for the project for a group, will you guys be selecting a project, or will we be selecting, or like will we choose people by ourselves?
So both are actually possible, but if you have some peers or friends that you want to work with, that is well and good. So you're already familiar with your peers. But if you would like for the team to, we usually get queries, but sometimes to develop a group, it takes time. Sometimes the timing might not be suitable. Sometimes the background might not be suitable to work on the project. So it's often recommended you can choose your own peers or friends. But if you want to work on a group project but would like us to, pair you up, you can just share your interest and our team can see if we can find a partner with you.
Okay, and will there be a chance of not getting selected for the research project after you fill out the form?
Sorry, I didn't get your question. Can you repeat?
Like, whoever fills the form will get the opportunity to do the research program, or you will have to fill the criteria, then you will be selected no matter if you fill the form.
Yes, so this is the form. This is a paid program. And for those of you who are attending this webinar live, you'll be, and also the participants who have registered for the webinar, you'll get access to this link. And you can fill out the details and our team will review. So we do get a couple of interest from the participants. Based on the experience and how convincing you are in the application that you're submitting to us, you'll be eligible for different scholarship opportunities. So that is how. I hope that answers your query.
All right, okay. Thank you so much.
Thank you. Any other questions? So I think we will be keeping this research scholarship form open till maybe for another 3 or 4 more days. So I hope you'll be able to fill out the form till that time. And we have shared the contact details in the chat box. So if you have any questions, you can let us know. And also we will email you the recording from today's session. So you can also simply reply back to the email thread with any questions that you have related to the research program or some of the opportunities that we provide. So thank you so much. And thank you, Namita, for joining with us today and sharing your presentation and your experience with the participants. And also thank you so much for all the webinar attendees for taking the time today to join and share your and attending it as well. So thank you so much and have a great day. So we will look forward to seeing you all in some of the upcoming research programs or some of the workshops that we conduct. And thank you so much and we wish you all the best. Thank you.
Thank you.